Starling equation used to calculate filtration rate, but simply knowing factors that determine filtration, we can suppose what is the reason of increased plasma filtration rate that clinically will manifest as ascites, edema, joint effusion, exudative pleuritis, pulmonary edema, etc. Basically, the general concept is that filtration occurs from blood and capillaries through the membrane, which is specific for every tissue, into the interstitial space. By interstitial space we understand also Biomed space and glomerulus, or intrajoint space in case of synovial joint. The composition of filtration membrane is different basically in every tissue. Usually membrane is composed of endothelial cells, basement membrane, and cells that are specific for a certain tissue. Endothelial cells and capillaries can be fenestrated or not fenestrated. Fenestration means that endothelial cells and capillaries have pores through which fluid and lipid-insoluble substances, as proteins for example, can cross into interstitial space. Such capillaries are located in tissues that have very high metabolic rate. For example, fenestration have liver capillaries that are called sinusoids, or capillaries in synovial membrane or in glomerulus membrane. Also, basement membrane can be present, or in some cases it can be absent. For example, glomerulus membrane has basement membrane, because the function of glomerulus membrane is not to allow proteins to enter into the Biomed space, because proteins are toxic for canalicular epithelial cells. Basically, it can be observed in crash syndrome, where myoglobin cross glomerulus membrane and enter into the canalicular system and cause severe damage to canalicular epithelial cells that results in acute kidney injury. Also a good example liver sinusoids, because in normal conditions they are fenestrated, and in pathological condition with inflammation, hepatitis for example, they lose their fenestration with simultaneous formation of basement membrane. And the reason is that liver has very high metabolic rate that requires high exchange between blood and tissue. And to facilitate this exchange, capillaries have pores and don't have basement membrane. And the lesser obstacles have nutrients on their way, the less time they require to reach hepatocytes, so the higher becomes their diffusion rate. But in inflammatory condition, capillaries lose their fenestration, and it's sort of protective mechanism to decrease pathogen spread and to protect hepatocytes. And also inflammation induces formation of basement membrane. It's basically an additional layer of connective tissue that is composed of collagen, proteoglycans and water. The same composition has cartilage, for example, so it's additional barrier, especially for lipid-insoluble substances as proteins. And with creation of additional barrier, filtration rate markedly decreases. And the third component of the membrane is cells that are specific for a certain tissue. For example, in glomerulus it's podocytes, in synovial membrane it's mostly fibroblastic synovial cells. So in kidney, glomerulus membrane is composed of fenestrated endothelial cells, basement membrane, and podocytes, and filtration occurs from blood and capillary into the Biomed space. For example, in case of synovial joint, synovial membrane composed of fenestrated endothelial cells and mostly fibroblastic synovial cells, and filtration is going from blood and capillary to intrajoint space. So it's the general concept how membranes are organized. According to Starling equation, the main driving force that determines filtration rate is difference in hydrostatic pressure between capillary blood and interstitial fluid. To explain this, let's take glomerulus membrane, its capillary endothelial cells, basement membrane, podocytes, and there is Biomed space. Blood and capillary has its own hydrostatic pressure, and fluid in Biomed space also has its own hydrostatic pressure. Filtration occurs in direction from fluid with higher hydrostatic pressure to fluid with lower hydrostatic pressure. And because blood and capillaries has higher hydrostatic pressure than fluid in Biomed space, filtration is going into the Biomed space. But what determines hydrostatic pressure? And actually it's mass of fluid. The shortest explanation without going into the details about types of pressure in the blood will be the more blood volume is accumulated in capillaries, due to whatever reason, it can be increase in inflow due to hypervolemia or hyperdynamic circulation, or it can be decrease in outflow with intravenous thrombosis for example, the higher become intracapillary hydrostatic pressure. And the higher become intracapillary hydrostatic pressure, the higher become difference in hydrostatic pressure, and the higher become filtration rate. And if hydrostatic pressure push blood out of the capillary, oncotic pressure keeps fluid inside the capillary and don't let fluid leave intercapillary space. So the opposite force to hydrostatic pressure difference is oncotic pressure difference between capillary blood and interstitial fluid. So again, there is capillary, membrane, and interstitial fluid. Oncotic pressure is formed by proteins, primarily albumins. And albumins, they are osmotically active substances. Literally, they keep fluid around them in intracapillary space and don't let fluid leave intracapillary space. And because proteins, particularly albumin's concentration is higher in capillary blood than in interstitial space, oncotic pressure difference becomes the opposite force to hydrostatic pressure difference. So the highest oncotic pressure difference between capillary blood and interstitial fluid, the lowest filtration rate. But there is such intricated parameter as protein, also called oncotic reflection coefficient. To explain this, recall that some membranes have capillaries with pores in order to provide more fast and massive exchange between blood and tissue. And exactly for this reason, some membranes don't have basement membrane. And the logic is that large pores and absence of basement membrane makes membrane more permeable for proteins, and in this case they can easily cross through this membrane. And the small pores and presence of basement membrane makes filtration membrane low permeable for proteins. And exactly this membrane permeability for proteins included into the equation protein reflection coefficient. Basically, small pores and presence of basement membrane makes filtration membrane practically impermeable for proteins, and this characteristic of membrane is reflected by very high reflection coefficient. For example, albumins in relation to glomerulus membrane have very high reflection coefficient that is close to 1.0, and 1.0 is the maximal possible value of reflection coefficient. In contrast to this, Levick capillaries called sinusoids have large pores in endothelial cells and they don't have basement membrane. So proteins can easily cross through these gaps in endothelial cells without serious obstacles. And because such membrane have very high protein permeability, a reflection coefficient of sinusoids for proteins is very low and is equal approximately 0.1. So the lowest permeability of membrane for proteins, the highest protein reflection coefficient. The highest permeability, the lowest reflection coefficient. And the question is, how permeability of membrane for proteins can affect fluid filtration rate? To explain this, let's suppose that some membrane have reflection coefficient 0.1. This membrane has large pores and there is no basement membrane. There is capillary with blood inside. So proteins in capillary blood through pores can easily cross from capillary space to interstitial space. And also let's suppose that blood in capillaries has high proteins concentration that gives it oncotic pressure 100 for example, and there is interstitial fluid that has low proteins concentration and oncotic pressure of interstitial fluid is 10. Recall that proteins are osmotically active substances, and because of that, they keep fluid around them. In this case, oncotic pressure difference between capillary blood and interstitial fluid, 100 minus 10 is 90, so it's huge oncotic pressure difference. And this means that initially there are a lot of proteins in capillary fluid that keeps a lot of fluid around them in intracapillary space, and don't let fluid leave intracapillary space. So potentially such huge oncotic pressure difference must result in very low filtration rate, because if hydrostatic pressure push fluid out of the capillary, proteins keep fluid near them and don't let fluid leave intracapillary space. So there is high opposite oncotic force to hydrostatic pressure, and in this case the difference between hydrostatic and oncotic pressure is minimal, and this potentially must result in minimal filtration rate. And everything that we already discussed remains as it is if this membrane is totally impermeable for proteins. But what if this membrane, as in our case, has high proteins permeability? And the principal question is, if this membrane have reflection coefficient 0.1 and is so permeable for proteins, does this huge initial difference in proteins concentrations makes a huge difference? If proteins can easily cross this membrane literally with water on them, and exactly this feature included into the equation low proteins reflection coefficient. And in this case, oncotic pressure difference, which is 90, multiply on protein's reflection coefficient 0.1 gives 9. So because this membrane is highly permeable for proteins, the efficiency of oncotic pressure difference dramatically decreases, and this results in increase in filtration rate. And if membrane is totally impermeable for proteins, all proteins remain in capillary blood, so oncotic pressure remains the same, it does not change, so it doesn't need any corrections. That's why in this case, reflection coefficient is maximal 1.0, and in this case 90 multiplied on 1.0 remains 90. So as we see in this case oncotic pressure difference remains the same. That's how reflection coefficient corrects colloidosmotic pressure difference. Another factor that affects fluid filtration is hydraulic conductivity. Hydraulic conductivity describes how easily fluid moves through the membrane. Basically, the main route of fluid through the membrane is intracellular contact space. Mostly it breaks in tight junctions. So for example, its synovial membrane, it has fenestrated endothelial cells and fibroblastic synovial cells, and there is capillary and its joint space. Fluid is moving preferentially through pores in endothelial cells and then through tight junctions in fibroblastic synovial cells into the joint space. And for example, with inflammation, damage to the membrane results in increase in quantity of breaks in tight junctions, as well as in size of these breaks. Breaks become larger and fluid flow through them increase. Because recall that flow is equal to pressure difference divided on resistance, and resistance is inversely proportional to the force power of radius. So the larger are breaks, the bigger is radius, and the lower become resistance. And the lower become resistance, the higher become flow. So with inflammation, membrane become more permeable to fluid, and is described by increasing hydraulic conductivity. That's how hydraulic conductivity affects fluid filtration rate. And the last factor is filtration surface area. Basically, the more number of capillaries are opened, and the more they are dilated, the bigger become filtration surface area, and the higher become filtration rate. So overall, hydrostatic pressure difference push fluid out of the capillary. Oncotic pressure difference corrected by oncotic coefficient keeps fluid inside the capillary, so it's the opposite force. Also, the more permeable is membrane to fluid and the highest filtration surface area, the more fluid is going out of the capillary into the interstitial space. So it's the general principle how filtration works.